Hey, welcome back. We're now in part 2 of the chapter 15 on innate immunity. We had just finished up with the body's first line of defense, the skin and the mucous membranes. Now we're moving into the second line of defense. So that means that one of these surfaces has been breached, either because there was a wound that an infectious agent got in through a scrape or something like that, or was introduced into the body. Now we rely on the next line of defense, the foot soldiers of the immune system. We're going to talk about a number of cells that play a role and some of the chemicals that play a role, most of them kind of floating around in the bloodstream. We'll do with the cells first, and then in a separate video, we'll go over the proteins in the blood. All right, so this is once the skin or the mucous membranes have been penetrated. There are cells, some antimicrobial chemicals, and some processes where these things interact with each other. Many of these things are contained in or originate in the blood. They actually originate in the bone marrow and then get into the blood and then find their way to the sites of infection. We're talking about the non-living part of this. We're going to talk about things like complement proteins. And then when we get to the third line of defense, we're going to talk about antibodies that are also floating around in the blood. But for the second line in the blood, we're talking about the complement proteins. The cells that we have in the blood are the white blood cells. These are also known as leukocytes. You've learned them before. There's 5 main kinds of white blood cells. And we kind of use this old-fashioned way of dividing them into 2 groups. The granulocytes, these are the cells that have granules in them. They look like they have grains, different colored grains in them. And then there are the ones that look smoother. They don't look like they have granules in them. Those are the agranulocytes. So A meaning not. And I'll tell you a little bit about them. All right, so these are the 5 different kinds of white blood cells here. The other 2 components, the other formed elements of the blood are the red blood cells and the platelets. But most of the cells we're talking about here for the secondary or second line of defense, we're talking about these white blood cells. The ones that look grainy, can you see in this cartoon? They look like they have little bubbles in them. Notice that the names all end in "phil," which means love, and it has to do with which of the stains they love, which ones they bind to or bind to them. These are the basophils, neutrophils, and eosinophils. And then the ones that we say are agranulocytes that don't have all of the bubbles are the monocytes. And these 4 are the big players here. This last one, the lymphocytes, those play a much bigger role when we get to the adaptive immune defenses, and that's going to be in chapter 16. So we'll stay focused primarily on these 4. The leukocytes, specifically the granulocytes first. They're called that because they were named before anybody knew what they did. It was just named based on the fact that the scientists that were looking at them through the light microscopes would, play around with the different dyes and color them differently, and they looked a little bit different. So these are the 3 kinds: the basophils, eosinophils, and neutrophils. Why they're called these things. This basic dye, meaning like alkaline dye, methylene blue, this loves that alkaline, that base dye. That's why it's called basophil, the basic loving. Then the acidic dye is the color red. It's the dye is called eosin, and so this one loves the eosin color, so it's called eosinophil. And then finally, the neutrophils are called that because it loves both the basic blue dye and the acidic red dye. And so the base and the acid balance each other, neutral. That's where those names came from. The way to remember them, they all end in "phil." They love something, and what they're loving is the different stains. And so those are the granules that stain. So those are the 3 granulocytes. Under the microscope, they look like this. The ones that we're going to care the most about are the neutrophils. Notice why. The vast majority of our white blood cells, we have 5 different kinds of white blood cells. I'd say half to 3/4 of the white blood cells of all 5 different kinds of white blood cells are this one kind of white blood cell, the neutrophil. So they're hugely important. The other 2 we'll talk about, but neutrophils are really the most important ones. Let's see what they do. The neutrophils are phagocytic. They eat other cells, and most specifically, neutrophils in particular focus on bacteria. The eosinophils are also sort of phagocytic, but they concentrate more on larger organisms like multicellular worms. The way to remember this is whenever people think about worm infections, they go, ew, worms. So, ew, worms, eosinophil for the EU factor. All right, so they phagocytize, that is, they try to consume the other organism. The eosinophils are too small to consume an entire worm, but they start to digest the worm from the outside. And they're capable of movement, diapedesis. They're capable of leaving the bloodstream. Basophils are not phagocytic. These cells do release some inflammatory chemicals in the bloodstream, specifically histamine, which we might talk about shortly. A little bit more about these leukocytes. We finished up with the granulocytes, and now let's look at the agranulocytes. There's monocytes. These are large cells that get even bigger when they leave the bloodstream. I'm going to pause for a moment. So the monocytes are the agranulocytes, the big ones. These are the ones that can leave the blood vessels. They also do diapedesis. They can change shape and they can leave the blood vessels and go into the tissues where there may be infection. Once they're in the tissues, they mature into what we call macrophages, which literally means big eater. And so they're phagocytic cells, devour foreign objects. It could be bacteria, it could be cellular debris of other kinds. Where neutrophils can eat bacteria, and neutrophils will stick around for a day or so. The monocytes that develop into macrophages, those are much, much larger. They can consume many more bacteria, and they stick around for much longer. And just in passing, I'm going to mention again that the lymphocytes, the other one of the agranulocytes, these guys are mostly involved in the adaptive immune system, and we're going to see what they do in chapter 16. But there is a small selection of lymphocytes that develop into the natural killer cells. Let's see. Let us pause again. But we're not going to make a big deal about these natural killer cells. All right, so here they are, the agranulocytes, the monocytes. They're not a huge proportion of the white blood cells, but they do have an important role out in the tissues when they mature into macrophages. And then we'll talk about the lymphocytes more next week. All right, a differential white blood cell count. So a complete blood count, a CBC, complete blood count gives you red blood cells and gives you information about hemoglobin levels and whatnot. But a CBC with diff, that's the differential white blood cell count. And we can get this information from the lab, from a patient's blood smear. And the kind of information that we can get from that is, one, is the white blood cell count elevated at all? That's leukocytosis, having elevated white blood cells. But then which white blood cells are elevated gives you a hint as to what's going on with the patient. So for example, if the neutrophil number is increased, and we already saw like half to 3/4 of the white blood cells are neutrophils, but if you get a sample back and it's 80% neutrophils, that should alert you to the fact that this patient is suffering with a bacterial infection. Or in contrast, if there's a huge increase in the eosinophils, that might suggest that this particular patient has a parasitic worm infection. This is a little bit of an aside. In this country, parasitic worm infections are actually pretty rare. And so what we found is that when we see somebody with an increased eosinophil count, that actually sometimes indicates that they're suffering from an allergic reaction, not a parasitic worm infection. And that's in fact one of the hypotheses for why we have so many allergies in this country, is because so few of us experience parasitic worm infection earlier in our lives. We have this crop of eosinophils just kind of cruising around looking for something to do, and that they intend to overreact to things, hence the development of allergies. But that is just a hypothesis. All right, continuing on, some cellular mechanisms. What do these cells do? I mentioned phagocytosis. It's a little bit of a complex process. I mean, it literally means the eating of cells, but there's this multiple steps to it. There's chemotaxis, which is super cool. These white blood cells can actually change shape and move toward chemical signals. So they like sniff out the bacteria. It's awesome. Adhesion, they grab a hold of the bacteria, ingest, pull it in. Maturation, the lysosomes have to fuse with this bubble where the bacterium is inside the cell, and start dissolving the thing, kill the bacterium, and then finally get rid of that waste. There's a little video here for you. Oh, chemotaxis. I'm going to pause this for a second. I'm going to stop my share because I don't think I shared with sound. Share sound. Let's do it again.
Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system.
Okay, don't worry about too much of the details of that. I just want you to know that these cells can actually change shape and they can move in the direction of particular chemical signals. Again, think sniffing dog going after prey.
The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization.
Okay, so that's another term I have not introduced. Opsonization, it's from the Greek, and it literally means to add condiments to. You can think of it as like to make tasty and make something more attractive to a phagocyte. The easiest way to remember this is that there are precious few words in biology that start with an O. And so opsonization, I think of that, it's like a wide open mouth. That's the signal that it's going to be attractive to somebody that's going to come eat it.
A process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes.
Okay, so then this thing gets broken down and it can't continue causing
After
infection.
phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation.
I'm going to pause that. So that little bit is going to be really important next week. The phagocytes eat things, but then they go and present them to the lymphocytes. So the lymphocytes know what they're looking for.
Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
Ta-da! So this is how our macrophages help us to get rid of bacteria and then go report about it to some other cells that are going to help out later. Ooh, here's some video. So that was, clearly a computer animation. This is an actual video of a light microscope of phagocyte in action. So you can see how this cell actually changes shape and chases after bacteria. It's awesome. I'm gonna get you. I'm gonna get you. Spoiler, it gets it. Soon. Yay, it's awesome. Yeah, so the cell itself is changing the cytoskeleton inside to continue changing shape and moving after the bacteria. Just great. Oops, that's not what I meant to do. Next slide. Okay, so here's a little bit of footage. Just savage. So this is just an animation. But the idea is that these giant macrophages are rolling around, going after bacteria in our bodies. Okay, so this is actually a light microscope. Digest. Oh, we can recycle the parts. That's kind of cool. So that's enough of that one. And then finally, that last little bit, the non-phagocytic killing. So we have the eosinophils. Remember the eosinophils, the worm infections. So they attack the parasitic worms, helminths, the technical term for these worms. They adhere to the surface of the worms, and clearly the helminth is like a really large multicellular organism. And so the eosinophil secretes materials that are going to start weakening the helminth to, kill it from the outside. And as we've mentioned, just like the neutrophil numbers being elevated is an indication of a bacterial infection, likewise, an increase in the eosinophil number is often indicative of either a worm infection, or as we mentioned previously, it might be indicative that the patient's having an allergic reaction to something. All right, so that is the end of part 2. I will see you over in part 3, where we will continue with the second line of defense. So the skin has been breached. Some of these cells are at work, but we're also activating something called the complement system, and that's what we're going to see in the next part of this lecture. See you over there.